Myself, Dr. Raktim Mukherjee, Assistant Professor and Head of the Department, Shri P.M. Patel Institute of Postgraduate Studies and Research in Science, affiliated to Sardar Patel University, Anand. The title of my today's talk is Biochemistry of Cancer. In this first video of the series, I shall introduce you to cancer at the very outset. Let's introduce ourselves to the world of cancer by beginning with the origin of the word cancer. What's the origin of the word cancer? The word cancer is derived from Latin word cancrum. This cancrum means crab. Already crab is depicted right in front of you. Further, cancer is a group of diseases characterized by, highlighting point is uncontrolled cell division. This uncontrolled cell division leads to growth of abnormal tissue. This abnormal tissue is also called as tumor. It has multiple layers, it does not show contact inhibition, and it grows in bulk and size. Now, based on this origin of the word cancer, let's understand what exactly is cancer. So, what is cancer? Cancer is a cellular tumor that, unlike benign tumor cells, can metastasize and invade the surrounding and distant tissues. What I will do, I will explain couple of terms. First, benign tumor cells. Benign also refers to non-malignant, and benign means it's localized to a specific area and it has not yet turned cancerous. Another term is metastatized, when cancer tissue which is localized does not remain localized. It spreads from one part to another and invade the surrounding or distant tissues, then we call it as metastasis. As long as cancer has not metastatized, it can be cured quite effectively. But once metastasis sets in, it's very difficult, and within several short span of time, the patient eventually dies. Imminent death is what the end result of cancer is all about. Now, what is cell proliferation? In the context of cancer, we always say that cell proliferation is enhanced in case of cancer and the cell escapes the control from cell cycle. So, what is cell proliferation? Cell multiplication or cell proliferation is a normal physiological process. This process is in response to injury. It can be an immune response. It can be a response for the cell that has died. So, after few cells die, the existing cells will replace all those dead cells. So, that also requires cell proliferation. It also replaces the cells that have been shed as a part of life cycle. Some of the examples are skin, mucous membrane, GI tract. These are areas which undergo sheer amount of mechanical pressure and continuous mechanical movement, and therefore, these tissues shed as a part of their life cycle. So, all these aspects are taken care by cell proliferation. Normally, the cell proliferation is always kept at balance. It is the impairment in this balance of cell cycle or cell proliferation that leads to cancer. We often hear the terms surgical oncology, oncologist, medical oncology. So, what this term oncology means with reference to cancer? Oncology is a branch of medicine that deals with etiology, diagnosis, treatment and prevention of cancer, wherein the word onco is a Greek word which means tumor. Now, what are the types of tumor? In this diagram, you can see some of the tumors, and as the diagram depicts, tumors can form even in the early neonatal period, as you can see in the upper half panel of the diagram. So, what are the types of tumor? First of all, the uncontrolled and rapid proliferation of cells can lead either to benign tumor or it can be malignant tumor. Benign tumor is non-cancerous. There is a mass of cell, but it won't metastasize at the later stage, whereas the malignant tumors are transformed ones which has undergone the several changes to be called as a complete cancer. So, malignant tumors are cancerous tumors. Benign tumors do not spread or invade other parts of the body or tissues, and benign tumors, you can live with benign tumors. The benign tumors are rarely a threat for your life. On the other hand, when you refer to malignant tumors, the malignant tumors can invade other organs. They spread to distant locations. This act of malignant tumors to spread to distant locations, as we discussed earlier, is called as metastasis, and this metastatic tumor often becomes life-threatening. Now, it's time for us to discuss the types of cancers, but before we discuss the type of cancers, the cancers can be classified by type of cell in which it originates, that is, where is the origin of this cancer, what is the cellular basis of cancer, where it originates, and also by the location of cell. So, basically it is the type of cell where it originates, and second, once the origin is known, what is the location of the cell which causes cancer. Accordingly, we have to refer to the types of cancer on the basis of the type of cell, the site or location of that particular cell, and the cancer it causes following transformation. Epithelial cell, the site or location is digestive tract, and the cancer is called as carcinoma. Blood cells, the site or location is WBC, white blood corpuscles. Leukocytes, leukos means white, cytos means cell. So, together leukos and cytos is leukocyte, means WBC or white blood corpuscles. So, blood cells, it causes leukemia, also called as blood cancer. Then lymphatic cell, the location is lymph node, and the cancer of lymphatic cell is called as lymphoma. Then we have connective tissue. The site or location appears to be bone. All cancers of the bone is given a specialized term that is called as sarcoma. You all might come across Ross sarcoma virus. Essentially, when you talk about sarcoma, we refer to the connective tissue, that is bone. Then comes melanocyte. Melanocyte is found in the skin, and the cancer that it causes is called as melanoma, the skin cancer. You all might be aware, students, that in hot summer days, when the considerable amount of harsh sunlight is there, you go out by applying certain sunscreen lotion on your skin. So, these sunscreen lotion acts as a sunblock where the sun rays can directly not invade the skin and its underlying cell types. Then comes germ cells. The germ cells are found in testes as well as ovary, and the cancer, that germ cell cancers are called as teratoma. Germ cell cancers are called as teratoma. Let's understand the etiology of cancer. In fact, cancer is multifactorial in origin. Let's understand, when we say multifactorial, let's understand what are the factors. Physical factors are present. The chemical factors are present, which acts as a mutagen. There are biological factors such as viruses. Many of the viruses are clinically proven to cause the transformation. Environmental factors. Environmental factors come under two categories. It can be a physical factor or it can be a chemical agent. Then you have hormonal factors, genetic factors, and mutation. Together, when you discuss the multifactorial origin in cancer, you have physical factors, chemical factors, biological factors, environmental factors, both physical and chemical agents, hormonal factors, genetic factors, and finally the mutation. Time to discuss the characteristics of cancer cell. Some of the important characteristics of cancer cells. The very first characteristic is uncontrolled proliferation. We always say in layman's language that cancer cells have become mad to the extent that they have left cell cycle regulation, never to return again. So it is uncontrolled proliferation. Then comes abnormal nucleus. The cancer cells have abnormal nucleus. You all know the genic machinery is in the nucleus. Further, the cancer cells lose anchorage. Generally, two types of cells are there: anchorage dependent, that it requires a substratum, and anchorage independent, that does not require substratum. So here the cancer cells have loss of anchorage, that is, it becomes non-anchor dependent. Further, the cancer cells are disorganized into multiple layers, and this disorganized multiple layers eventually lead to loss of contact inhibition. So we always say that those cells where there is a disorganized multi-layer phenomenon, we say it has lost contact inhibition. So cancer cells also have a property that is called as inhibition of contact inhibition or loss of contact inhibition. Further, cancer cells form tumor. It undergoes metastasis and angiogenesis. Metastasis, of course, everyone knows. Just now we discussed one cancerous cell traveling to lymph node or any other route and infecting the surrounding cells. It also undergoes metastasis and angiogenesis. Angiogenesis is formation of new blood vessels. Further, the characteristic of cancer cells, the cancer cells lack differentiation into specialized cells. The cancer cells also increase the amount of anaerobic glycolysis. Now, what are the biochemical changes associated with cancer cells? There is an increased synthesis of DNA and RNA. There is increased rate of aerobic and anaerobic glycolysis. I will highlight aerobic and anaerobic glycolysis. This rate of increased anaerobic and aerobic glycolysis is essentially to meet the increased demand for energy for more cells. If the number of cells are more, it will affect your energy reservoir. So, these are the biochemical changes. Finally, the rate of anaerobic glycolysis is tremendously increased under the influence of cancer cells. What is the basic standard cancer management? There are various ways and means. One, of course, is surgery, second is radiotherapy, and third is chemotherapy. We will discuss each point separately. Surgery is surgical removal of cancerous tissue. Radiotherapy is destruction of cancer cells using radioactive rays. Chemotherapy is killing of cancerous cells using drugs. Some of the examples we see in the last section, you have cisplatin as one of the important analog of So, let us discuss what is surgery. Surgery is removal of cancerous tissue. Radiotherapy. Radiotherapy involves destruction of cancer cells using radioactive rays. Chemotherapy kills cancerous cells using drugs. Now, what are the drugs used in cancer chemotherapy, or can you name some anti-cancer drugs? We have quite a few. First is methotrexate. Methotrexate, the chemistry of the drug is, it is a folic acid analog, and therefore it inhibits dihydrofolate reductase. Reduction in dihydrofolate reductase will lead to dihydrofolate reductase will allow the methotrexate to remain in circulation for longer duration. Then you have 6-mercaptopurine. As the name suggests, 6-mercaptopurine is a purine analog. This 6-mercaptopurine inhibits formation of adenosine monophosphate, that is AMP. Then 6-thioguarine, it is also a purine analog and it inhibits thymidate synthesis. Mitomycin C is an antibiotic. This links DNA base pairs. Actinomycin D is also an antibiotic. It inhibits transcription. And cisplatin, it's a platinum compound and it forms DNA adducts. Vinblastine and vincristin, these are alkaloids from rosy periwinkle, and vinblastine and vincristin inhibits spindle movement. Now, we will discuss some of the naturally occurring anti-cancer substances. Vitamin A and beta carotene, these are antioxidant vitamins. They mop up the free radicals. Vitamin E tocopherol is also an antioxidant. Lycopene, a carotenoid present in ripe red tomatoes, because of its antioxidant action, is a very potent anti-cancer substance. Ascorbic acid, vitamin C is an antioxidant. Selenium, a trace element, has anti-cancer activity. So, zinc is also a trace element. Now, some of the agents which were not present in the plasma. Quercetin is a flavonoid present in apple, and this is the reason we always say an apple a day keeps the doctor away, because quercetin is a flavonoid present in apple, and this apple has anti-cancer activity. So, quercetin is a flavonoid present in apple which is anti-cancer. Then you have glucosinolates. Under glucosinolates, quercetin of course is shown to prevent growth of prostate cancer cells. Glucosinolates present in bitter Brussels sprouts, and this glucosinolates contain sinigrin. Sinigrin suppresses the development of precancerous cells, and glucoraphanin, which breaks down the compound which is called then you have sulforaphan, which neutralizes carcinogen. You have the most common towards the end, epigallocatechin gallate, that is EGCG, epigallocatechin gallate. Remember, this is found in green tea. Epigallocatechin gallate, EGC is found in green tea. Now, what are the physical agents for cancer? X-rays, gamma rays, UV rays. And what are the chemical agents? Asbestos, it causes asbestosis. Aflatoxins synthesized by the fungi Aspergillus flavus. Aniline, which is a dye. Nitroso compounds. Coloring agents, benzoapyrene, and polycyclic aromatic hydrocarbon, that is PAH. Some of the modification can be based on lifestyle changes. You have to quit tobacco chewing, smoking, as well as alcohol. So, lifestyle changes, tobacco chewing, smoking, and alcohol. Now, we will go to experimental carcinogenesis. Having discussed the chemical agents, now let us understand the two-stage chemical carcinogenesis. First, I will represent the important part with this cartoon, and after that we will come to the original topic. So, suppose this particular mouse has to be made to a tumor free mouse or tumor bearing mouse. What you can do? You can take the mouse, mutate the various genes, so first you have initiator, and after that you have tumor promoter. If both initiators and tumor promoters are present, only then so normal mouse, first of all you give initiator, and after that along with initiator carry out mutation, it becomes a tumor bearing mouse. On the other hand, there is no initiator or no tumor product, then the mouse that develops will be tumor free. So, what is experimental carcinogenesis? Neoplastic transformation can be produced by chemical, which is a dynamic multi-step process. The best example that we can give is also the formation of skin tumor in mice. When you talk about skin tumor in mice, there is a chemical benzpyrene. If this chemical is given alone, there is no tumor that develops. Whereas when benzpyrene is followed by several applications of croton oil, many tumors might develop. So the question that arises, why benzpyrene alone do not cause tumor? Question 1. Question 2, what is so special that is happening when you applied the croton oil? In fact, many tumors have developed. What might be the reason? We continue with experimental carcinogenesis. Application of croton oil alone without pre-treatment with benzoapyrene, no tumor takes place. So only croton oil also there is no tumor. So what is the conclusion? The conclusion is two definite stages can be observed in this study. One is initiation, another is promotion. So two different stages. One is initiation and another is promotion. Both studies can be there. Under initiation, benzpyrene is rapid and irreversible, causes rapid and irreversible modification of DNA, and later on croton oil, which acts as a promotional material. The croton oil application is a slower process. Based on all this, let's understand what is mutation. There is sudden change in chemical structure of the DNA. That aberrant change is called as, in chemical structure, is called as mutation, and the substance which causes mutation is also known as mutagen. Some of the examples of mutagen are X-ray, gamma ray and UV rays. So what happens during mutation? There is a sudden change in the chemical structure of DNA. This leads to aberration in chemical structure. This substance which causes mutation is known as mutagen. Example of mutagens are X-rays, gamma rays and UV rays. Now, what is oncogenic virus? The viruses which cause cancer are known as oncogenic virus. These viruses can viruses can integrate their gene into the host DNA, and after integrating, they will take over the entire regulatory mechanism for the host cell. The host cell will produce viral genome instead of host genome, and because the host cell has already been transferred, or it has already been transformed cell. This transformed cell leads to uncontrolled multiplication of the host cell, also called as transformed cells. These viruses can integrate their gene into the host DNA and take over the entire regulatory mechanism. The host cell will therefore produce viral genome instead of host genome, and this is called as transformed cell. Ultimately, it leads to uncontrolled multiplication of host cell, that is transformed cells. Let's understand the two steps, that is initiation first and followed by promotion. So, first we will go with initiation. Two definite stages can be observed in the experimental study. The first step that is observed is initiation. There are 2 points to be observed under initiation. First, the stage of carcinogenesis caused by application of benzpyrene, it is called as initiation. Benzpyrene is thus called as initiating agent. The next step, of course, is promotion. So, what is promotion? In the stage of carcinogenesis caused by application of benzpyrene is called initiation. The second stage appears to be rapid and irreversible. This involves the induction of certain irreversible changes in the genome of the cells. What these changes mean to us? Essentially, it brings about modification in DNA resulting in one or more mutations. What are the characteristics of initiated cells? Some of the characteristics of initiated cells are that the initiated cells are not transformed cells, is very distinct from transformed cells. The initiated cells do not have growth autonomy, that is, they are dependent. Unlike normal cells, the initiated cells give rise to tumors, and these tumors are given rise when the initiated cells are stimulated by promoting agents. After initiation, now let's understand what is promotion. The second stage of carcinogenesis is called as promotion. This promotion results from the application of second agent, the croton oil, and this is called as promotion. Alone initiation would not give rise to carcinogenesis. We always require promotion for the same. Croton oil is just a promoter or promoting agent. Promoters cannot produce initiation. Promoters bring about. So, promoters cannot produce initiation. They just bring about the tumor induction in the previously initiated cells. Most chemical or carcinogens are capable of acting both as initiator as well as promoter. Let's understand the biochemical mechanism of action of croton oil as a promoter. This concept is very important for students who are about to appear for Sardar Patel University external examination. Active agent present in croton oil is a mixture of furbol esters, the most active one is TPA, chemically TPA is 12O tetradecanoyl furbol 13 acetate. Most of the common site of attack is guanine and the addition of various carcinogens to N2, N3, N7, O6 and O8 atoms of the base. Which base? That is guanine base. This process is critical to carcinogenesis and the alterations in two types of genes. One is proto-oncogene, another is recessive cancer genes. Both these types we will study in the second segment of the video. The sequences encoded by these families of genes regulate normal growth and difference. Thank you for viewing this segment of the video. Do like and subscribe for more videos in the same series of Biochemistry of Cancer.